thank you. I know everyone is super excited to hear about a database. So that TV is off, so I'll have to read here. So IMGVR4 is a database of viruses genomes within a framework of functional, taxonomic, and ecological metadata. That's the official title. My glass is foggy. But I think that a more down-to-earth name would be, we identified viruses in tons of samples, and now you don't have to. So I'm pretty sure every single one of you either read or even written the following phrase: viruses are the most abundant biological entities on Earth. Yeah, I've read it several times. And there is a reason for this. This is true. Viruses are super abundant. And in the last couple of years, we've been trying, to understand this abundance, this diversity. We barely scratched the surface. So in 2016, the VPIs here from the JJ and some other JJ folks, they set out to uncover the diversity of viruses in all of the IMG metagenomes. So using a set of marker proteins, they unveiled more than 125,000 phage genomes, which expanded the number of virus proteins by 16 times at a time. So they had this amazing resource in their hands, but this raised the question how to share this with the community, right? Because, well, people should have access to this. And that's when IMGVR came to be. So again, it's a database of our genomes that is integrated to the IMG ecosystem. I'll tell you later in the presentation why is this so cool. It provides multiple layers of information like function annotation, environment, taxonomy, host, and much more. And it also provides you with tools for standard, sequence search and metadata search. Well, and that's it. We've been updating it constantly. So first release 2016, second release 18, third release 2020. Everyone knows what comes after 2020, 2022. That's what I'm talking about. And yeah, there was a huge increase from 2016 to 2020. But in the meantime, other papers also came out, exploring this viral diversity and environmental viromics. They explored different parts of the virus sphere. So single-stranded DNA viruses, RNA viruses, gen viruses. And they not only increase the total number of genomes, but also push the boundaries of the virus sphere, what we know are viruses. And the IMGVR identification pipeline didn't really capture all of this diversity because it was one of the first big screening of viruses. It wasn't really prepared for this new wave of viral groups. So we needed to update that. And last year, we developed GeNomad, which is a tool that allows large-scale identification of viruses, also plasmids. It's a shame that I can't talk about plasmids today, but yeah. It covers much more of the diversity of the virus sphere. It allows for systematic identification of proviruses, integrated viruses. That's something that we couldn't do before. It is really fast, which was needed because, as I said before, we are analyzing billions of sequences. We need something that is really fast. And it has a couple of other additional perks, one of them being taxonomic assignment, which is always really useful. And finally, it is available to the community. The previous pipeline wasn't, so anyone can go in there, please go, and you can use to analyze your own samples. It also is available as a web app in the NMDC Edge platform, so even if you don't have experience with bioinformatics, you can use it online. I don't really have enough time to talk about it. It's not the goal of this presentation, but one thing that you should know is that we are using it because it is really precise. We did several benchmarks, compared it to several tools across different length ranges. This is one of the benchmarks that I'm showing here, because I just want to highlight that it outperforms other tools across different sequence length ranges, which is important when you're analyzing metagenomes and you have very fragmented metagenomes with short sequences. Maybe you want to find the viruses in there. So that's one of the reasons that we've been using. Okay, so now, by the beginning of this year, we already had geNomad. It was functional, and we applied it to, again, thousands of prokaryotic genomes, metagenomes, metatranscriptomes. And well, I'm showing that bar plot again, as you guys can expect, because now the next bar will be super big and I want to make everyone super impressed. And yeah, that's the case. The bar is super big because again, going back to the beginning of the presentation, viruses are the most abundant biological entities on Earth. So the total number of virus species, so this is, the numbers here correspond to VLTUs. They're like species level sequences. So the number of virus species is now 10 times larger. It went from about 900,000 to more than 9 million different species. Even if you only take into account the high confidence set of sequences, I'll talk what the high confidence set is briefly. But even if you only take that into account, the number of virus species is now 3 times bigger. And some cool things are like, now that we have a systematic way of finding proviruses in genomes, 1.2% of all of, 2.5% of all of our sequences are proviruses. This is really cool because you have a direct link with the host. And also, the previous pipeline wasn't really executed on metatranscriptomes. Now we did. And about 7% of all of our viruses came from metatranscriptomes. And a good chunk of those are RNA viruses, which I'll talk briefly. Okay, going back to that high confidence set. So the vast majority of IMGVR is just based on automatic prediction of viruses using geNomad. And then we have roughly 2 types of sequences. We have the sequences whose viralness is well supported. It is either very similar to a reference viral genome, or it encodes tons of viral hallmark clonings. But we also have some sequences whose viralness is not that well supported. They're most likely viruses, but maybe they encode few hallmarks or no hallmarks, or they don't really have any detectable similarity to a reference virus. And we have different types of users that are interested in those. We have users that want to be conservative. They only want to look at the well-supported viruses. And we have users that are interested in just looking at every single thing in the database. So to make everyone happy, we just stratify the database into 2 different categories: the high confidence viruses, the ones that are very well supported, and the putative virus, the ones that are less supported. This is a very conservative estimate. Yeah, so more than 30% of the database, more or less, is from high confidence virus. I talk really loud. Oh, sorry. Okay, going back. The high confidence viruses were further classified into high quality, medium quality, and low quality. Those categories are based on their estimated completeness, which we measure with CheckV. So high quality viruses are viruses that are, whose genome is almost complete, over 90%, medium quality between 50 and 90, and low quality less than 50. So we also provide this estimated completeness for users so they can filter the way they prefer. Okay, let me use the microphone now. Because geNomad provides taxonomic assignments, and we also perform comparison with reference genomes, we managed to assign 90% of the sequences to a viral taxon, a CTV viral taxon. This not only is useful for the user, but also provides us with some interesting insights. So as expected, this is not really a surprise to anyone. Tailed phages comprise most of the database. So among the high quality viruses, they are about 73%. But we saw a big expansion of other types of viruses. For example, single-stranded DNA viruses from mono-DNA virion. They now are 7% of the high quality viruses. Ribovirial RNA viruses, though the expansion was even bigger. Now we have about almost 20% of our database now of the high quality viruses in the database is RNA virus. This is a 47-fold increase over the previous version of the database. This is not only interesting, but it is really cool that it is a consequence of all of the recent studies in environmental viromics that really push the boundaries of the virosphere. Another information that we provide is host. So we assigned host bacteria and archaea hosts for about 7% of the viruses. We did this using different approaches. One of them is a guilt by association. When we found a virus in a host isolate genome, we can directly infer that that genome is the host. We also use CRISPR spacer matches, which is something that was discussed yesterday in several presentations. And Camargo matches to a big data set of prokaryotic genomes that we had. We also provide for the users a BLAST interface for our own database of CRISPR spacers. So if you have one genome that you want to try to infer the host, you can just blast it against our database. And the last layer of metadata that I'll talk about today is the geographic environmental metadata, which is probably the ones that is the most unique to IMG. So all of the viruses that were identified in metagenomes or metatranscriptomes, they are associated with the geographical coordinates of that sample and also the environment of that sample. The samples are classified into a ecosystem classification. So for example, you will know if that virus came from a freshwater environment, a soil, a human-associated sample. And this comes in handy for ecological questions. If you have an ecological question, you can use that. But also, it allows us to state the obvious. The virosphere is still largely underexplored. When you look at this globe, the vast majority of the samples came from the same place. The places in the Earth that harbor the most biodiversity, they're still underexplored. Also, when you look at the rarefaction curves, we're nowhere close to the plateau, to exhausting the diversity. So there's still much more to uncover. Okay, going back to the database now, that's why I'm here. Users can use all of those features that I've been talking about to find viruses that are relevant for their own research. So they can query viruses according to all of those things that are in the screen that I've talked about in this presentation and some other additional ones that I couldn't. And you can find things that you want to. For example, let's say that I want to find high quality genomes from Press viralis, so press phages that use alternative genetic code are found in a human gut metagenomes, infect bacteria, and encoded terminates. You can just go to the interface. You can choose metagenome, genetic code 15, that's a UGA codon reassignment. I don't have my glasses, but I know that there is a human gut in there in the environment. Taxonomy, you can put the terminase PFAN in there, so you only find phages with a terminase, and that's it. So then you can browse the results. In this case, I got 67 pages, if I'm not mistaken. And when you find something that you're interested in, you can dive into it and look at the GMap, for example. That's most of what I wanted to talk about today. The cool thing is that it's already online. You can go to the website right now and just do whatever you want. There is this form for questions. The slides will be available, so you can just grab the link later. Again, geNomad is also available. You can use it. You can use it as a web app in the NMDC Edge platform. And that's it. There were lots of people involved in this database. Everyone contributed to it. I can't, put together a website with a database. I really wanted to at least mention Simon, that was involved throughout the project, helped me to put the data together. Krishna, that coordinated with me all the database part of things. Stephen, that also helped me with data, and Mikos, he's our boss, so he needs to be here, but also he coordinated everything, and he is the one that put everything together. Also Amy, that is the general coordinator of IMG and also got her hands dirty for this project. That's it.